So thanks for this opportunity to talk to you. How many here are synthetic organic chemists? Okay. I'm going to talk to you. Yeah. I'm not going to talk synthetic organic chemistry. I'm going to show you sort of things at an impressionistic level, give you an idea what the capabilities are. Some jargon. Hits are what you get, obviously, from screening. But probes, these days, it carries an NIH definition. You know, we don't be too ambitious. We don't want to say drugs. Drugs are things we put into people. Very, very demanding. We're going to say something about that at the very end. The criteria for making something a probe, however, depends on how you get it. If you've got purified components of, say, some enzymatic system, some pathway, the NIH was defining it as something better than 100 nanomolar potency. If you have a cell-based assay, 1 micromolar would have met their definition. It indicates that there's a gap between what you do in test tubes and what you do with cells. And cell-based assays have certain advantages. If you're seeing a signal in a cell-based assay, it means, hallelujah, I'm very likely to have a signal in a biological system. On the other hand, if it's a phenotypic assay, you don't know exactly how you did it. And so these are the themes that are going to appear in 2 projects I'm going to show you. Now, the hits suggest that I'm giving a talk for a high-throughput screening facility. Okay? But just as you heard, you don't need to be doing high-throughput screening to use the facility. You don't need to be doing screening to use the chemistry. You can get access to chemical entities in a variety of ways. Good to remember that if you use a library, even if you buy the library, you don't own the compounds. If you assemble the library from commercial molecules, you certainly don't own the compounds. And so another concern, often looking in the background, is how do I get something that's unique? Translate, how do I get a new composition of matter that might be patentable? Okay? That's an intellectual property issue. You've got to get somehow out from under patent space. So even as you're reviewing the literature, you also, the chemical literature, the biological literature, you have to be reviewing the patent literature to see just where you stand. And that's something we do on a parallel basis. And that's something where Deng will frequently generate a patent summary report, give you an idea of where we stand relative to other patents in the field. So with that as overview, let's begin. I'm going to show you 2 projects. One actually doesn't begin with high-throughput screening, but it illustrates a number of features. Just want to go through it quickly. This was a project in chronic pain. Richard Ambron in pathology had discovered, and his associates discovered at the end of 10 years of NIH-funded investigation, that activated PKG1 alpha materializes in severed or inflamed axons. It gets transported retrograde to the DRG, the dorsal root ganglion, and at that location turns on genes which then have effects upstream and lay down the pathways for chronic pain. Problem? Getting something selective and potent for PKG1 alpha. Well, it turns out there was a molecule in literature, Valinol, not a very appealing molecule to a chemist. Unusual ring sizes, lots of polar substituents, but very, very potent. Very low nanomolar potency, 2 nanomolar potency. Unfortunately, not selective. PKG and PK both inhibited with single-digit nanomolar potency. But we could look at an X-ray structure in the literature of Valinol and the PK binding site. We could look at a homology model for PKG and look at how Valinol might bind. We could look and see where the stresses and strains were, how we might make modifications that might get some selectivity. And then we need some molecule. So this is the structure. You can imagine breaking it here, breaking it there. Now you've got a benzophenone piece, a hydroxybenzamide piece, an azapine piece, and then you could start making these things. And if you had all of them, you could begin to assemble them. And so this was actually, we prepared this, we put it together, we made Valinol. Okay. This is optically active. We then wanted to construct varieties. I'm going to give you an idea of how we do this. But just basically, we have these 3 components. It's convergent synthesis. They all come together. So we can make a variety of ring structures. Maybe not just 7-member ring. Let's try 5. We try 6. Try 7. We can use different building block number 2s. We can use different building block number 3s. Now we can start making all sorts of things. So this is the synthesis of building block 1. And this would be a synthesis, for instance, of a 5-membered version, not the 7 that appears in the natural product. And you begin with an optically active material, and you end with 1. And we can have ester linkages, or we can have amide linkages. I'm showing you the amide. I'm giving you just one arbitrary glimpse into the chemistry. For building block 2, this was not the hydroxy benzamide version. This is the indene. Number 3, this is a stripped-down version of the benzophenone. Okay? And again, you get a feeling for the magnitude of the chemistry. This could take, first time out of the block, 2 chemists 3 months to play with this and tease it apart and get it to work. So it gives you an idea of what you're asking for when you say, can you make this? Sure. How much time? How many people? We assemble the building blocks, and we get a molecule, B33. Okay. So let's look at some SAR. Here we can look at different ring structures. This is the 7-membered ring, the central ring of Balinol. We can look at 5. We can pull out the nitrogen. We can have 5 with the nitrogen. We can also look at a variety of structures where attachments are occurring off nitrogens. And we see that in terms of activity, when you do something like this, you lose it. Okay? We start making these changes, and we maintain potency, but we don't get any selectivity. Okay? But it's easier to make a 5-membered ring than it is a 7-membered ring. So we've simplified the problem. Okay, so we've taken something that would be really, really difficult and almost impossible to imagine on a large scale, translate, say, 20 grams, so you could do animal studies after having demonstrated something in a test tube or at the level of cell culture. We could also look at the benzophenone, and we could try simplifying that and strip these down. And we again had some guidance from the computational biologists that we were collaborating with. We can do unusual things at Columbia because Columbia is a large, diversified enterprise. There was a company that started from Columbia professors, Columbia Technology, called Schrödinger. And Schrödinger does calculations, it's computational chemists, and they were the ones who did the docking, and they were silent partners. So if we were to talk to, say, a company later on, the company would never know Schrödinger was involved. Schrödinger just gets a little slice of the Columbia royalty. Okay, so this is something that you could bring to bear if you actually understood the details, the molecular details of your target. So we had them model this, and they thought this would be good. We can look at a variety of interactions using this benzophenone. Okay, now we're using the 5-membered ring that we have validated as accepted. A quick note, there's an assumption that's made. The assumption is if you determine that this can be changed, because you keep everything else constant, you change this little piece, and then you keep everything else constant, and you change this little piece, that you'll be able to put them together. That is a hypothesis. Chemistry is a very empirical enterprise. Okay? It is more likely true than some random change, but it is not something that you can take to the bank. Okay? So this is still very empirical. Therefore, it can take an unreasonable amount of time when your ideas turn out to be false, and you've got to go back to square one and try new changes. Maybe one that was overall less effective will in fact be more compatible. And then you'll see additivity, if you're really lucky, synergy. So you can look at a number of changes that are made here. And there are some changes. We start with this. A stripped down species, a bit simpler. We've lost a little bit of activity, but 70 nanomolar could be just fine. But again, no selectivity difference. Modeling suggested that if we had connections off of here, that might be accepted in the PKG docking, but not a pKa docking. And so you can see that when we put a 3-carbon chain, okay, convert this hydroxyl group, this phenol alcohol to an ether, okay, we lose a little activity, but we really kill pKa. Okay, and this is how we start to build in selectivity. We can make a variety of changes. You can put in 2 oxygens. You can start making it longer, 2 oxygens, but an extra carbon bridge. And now we're starting to kill pKg. So there's a limit to which a chain can be tolerated even for pKg. But here we've got 40 nanomolar plus 1 micromolar pKa might be acceptable. You'd have to eventually try this in, say, animals. Concern is a cardiovascular side effect, hypertension. So you could try this. If you could see a signal in a pain model and not see hypertension, then you've got enough selectivity. You can never have enough, but 500-fold would be a reasonable goal. Obviously, this one doesn't quite make it, but not too bad. Other changes. This is an ester, and esters are labile. Amides would be superior. Here we see a diamide, and it's accepted. but no change in selectivity. On the other hand, if you had a diamide, and which we'll be coming up to shortly, with a side chain, maybe you could get stability and selectivity. And here are some of the diamides. And we can look through and we start to get into the high B28, B29 series. 7 nanomolar for pKg, 5 for pKa. Okay, 28 has the chain out here on the benzophenone, and has an amide. So it's going to be a diamide. And this is just a summary of some of those changes. Okay, if we went from the ester amide to the diamide. We could put this into animals. I'm just going to cut to the chase here and tell you that we've made, at this point it was 60, actually we've made about 90 now. Nanomolar potency, high specificity, up in the 5+ micromolar range for pKa. It's been tested against a panel of 280 kinases, and it is most selective for pKg. There are 4 other kinases out of the 270 that are close. Whether they are too close for comfort is something that you can only know in the animals. So far, the animals seem to be tolerating it. Let's switch gears.
Doing fine.
Okay. Let's switch gears. This is the use of a high-throughput screening facility. It was actually the Columbia screening facility. It was the form fruit of what exists today. Jim Rothman wrote the grant. He was the biologist. We were the chemists. It was the MLSCN, NIH pathway approach. It's something that they're slowly or rapidly retreating from, depending on your perspective. And this was an assay for NF-kappa B. It was the translocation. It was a fluorescent tag, GE whole-cell screening, and we could look at NF-kappa B moving from the cytosol to the nucleus. And a number of hits appeared in the screen. So chemists looked at these hits.
Were you looking at inhibitors or activators?
Inhibitors.
Yeah.
We were inhibiting NF-kappa B from going to the nucleus. Did I say activator?
No.
Oh, okay, I didn't.
Until you say that.
Yeah. I'm a chemist. I'm not concerned. I just think I'm optimizing the signal. Okay, so we have a series of hits, and when a chemist looks at this, and perhaps even a non-chemist, you might not want to attack these. Okay, this is just off the chart. Okay, this is a Department of Chemistry, 2 or 3 postdocs, 2 years, maybe 3. Okay, long stretch. The chance that you're going to simplify this to anything useful, pretty slim. This is an open ring structure, hard to control, hard to predict what's going to happen with each change. Not a good choice. This might not be so bad, but overall, there's a little bit too much functionality. We might strip it down. We might not be able to strip it down. We got lucky with Balinol. All things being equal, we might not want to play with this one. So now we're in this series, and this is a symmetrical molecule and didn't look too promising. And in the end, we focused on these. One thing you should think about with screening is counter-screening. So one of the things they had was the capacity to do very high-throughput counter-screenings against a variety of targets to see if they had unusual activity against things you wouldn't want. Okay? And this could be done for compound 4,5, compound 09. And we could eventually go back against that group of 4. And that's, in the end, how we identified the 04 and the 09 compounds. They didn't have too much of this ancillary off-target activity, we hoped. So here are the ones we went after. One thing you want to do is see what's commercially available. So when you get a hit, you immediately go to the literature, you go to the company, you look at the chemical catalogs, and you buy every molecule that's similar to it, okay? Because it's a lot cheaper, a lot simpler to buy molecules than it is to make them. So you buy all these. You go to the parent library and see if there are other molecules you didn't test. Maybe you tested only a subset. You tested 50,000 compounds. In the 100,000 library, there's still a few more. So you pull those out. It's called a cluster analysis. It gives you an idea of where you stand and gives you some cheap SAR, right? Structure-activity relationships. And so there were some out there that we could look at. And there were some others. But we could do the same thing for the 4-5 series. Now a reminder that this is a cell-based assay, okay? And with TNF, we see this trans location, and we're looking for compounds that would block this. And one slide slightly out of order. We'll come to it in a second. It also means, I just want to remind you, that it means we don't really know how it acts. We don't really know where it acts. That's going to become a separate research project. Okay, as you see, a potentially daunting one. So this was the 09 dissection, and we can take this and we can break off this piece. We can remove the nitrogen. We can come up with a series of simpler versions. We're going to try and figure out what is the business end of this molecule, or is it all business? Is it all necessary? Okay, you've got to come to terms with it. So you just begin taking rings off, compressing rings, changing it, and it's completely exploratory. Each one of these a completely separate synthesis. Okay, so a lot of work as you're trying to narrow down what it is you're actually going to make. Once you're going to make one thing, you can try a synthesis where there are branch points, where you can get 20 versions of this and 20 versions of that. Okay, and then you begin to cover some space. But you've got to narrow it down. So the first step is figuring out what the business end is. So let me just leave the 09 series here and tell you that all of this effort and a lot of molecules made, and nothing came of it. Okay? We just didn't improve any activity, couldn't really dissect this thing, couldn't make any progress. Okay? This sometimes happens. Now that's not to say we couldn't make progress. Remember that Merck, when they are making a molecule for medicinal purposes, will put a chemist per carbon. There's 20 carbon, 20 chemists working on it. They don't each work on their own carbon, right? But I just want to give you a feeling for the scale. Here you've got maybe one chemist on this project, or two chemists on the project. So they're going to look around and see if there's anything definable. When it didn't look promising, we just dropped it. We went to the other one. Doesn't mean that there isn't a Nobel Prize on that previous slide, but we just don't have time to work on it. So we went to 4-5, and we looked to see how this might be played with. And is this nitrogen necessary? Could you have more than one? Is this ring system necessary? Could you break this bond and give this a little bit more motion? This is a little bit floppy. Potentially, it would be a lot more floppy if you could break this and it still retained activity. That would be a lot simpler to make. So now we could just make this bond, and we'd have brought these two together. You actually don't need to have this. And so this is, again, how we begin to look at various possibilities. Get rid of the sulfur, put in phosphorus. These are the ideas. Very exploratory, very empirical. And so we could look at a variety of substituents on this open ring structure. We could also look to make the closed ring structure. And we tried a number of standard routes. They failed. So Yuli Z, very talented chemist who is with us. He's now, is he running a company now? He's in China. He's running China right now. And we're still hoping he'll return at some point. Was able to figure how to do this. It becomes a separate paper, in a chemistry journal. So if we have to create a little new chemistry to get something solved, it can be done. And so we look at a series of molecules. Oh, this is the slide that I could have put a little earlier. This is, but maybe it wouldn't have made complete sense. We could look at a variety of these molecules. We could look at potency, and we're starting to get sub-micromolar. This is a cell-based assay. We met the NIH definition of a probe. We didn't try and optimize them further. Okay, this project was satisfied when we reached something that was 500 nanomolar. Okay, those were the terms of engagement. But we were also concerned with, and this should be enough to demonstrate activity in a variety of disease models. So I'm going to show you one in just a second. But we also tried to figure out what's it acting on. Well, we looked at a variety of kinases, all negative. We looked at a variety of pathways in the cell. We just could not determine. And to this date, we have a hypothesis. And we're exploring that hypothesis. But we don't exactly know how it's acting. Just know that it does what it does potently. We also had a collaboration going with the NIH. Since they were screening the same library using a different type of screening, they came up with similar compounds. But they weren't actually able to make the 4-ring system, because they tried the standard chemistry and it also failed. So we combined forces. So we had our chemistry and their open ring chemistry, and it led to a publication. So these were our best out of this effort, and the MLSCN and the NIH data were pretty similar. So the two different assays, but similar results. So we were pretty confident in this. And again, the NIH was happy to stop at this point and make some of these molecules generally available. But then we developed a collaboration with Owen O'Connor. Owen O'Connor is a member of the Cancer Center. He's a lymphoma expert. And he was interested in different types of lymphomas, and there are different types depending on whether it's germinal or non-germinal. And he had various cell lines, and we could begin to screen. And some of these cell lines are actually addicted to the NF-kappa B pathway. That pathway has to be active, has to be continuously, constitutionally active for these cells to thrive. You block the pathway, they die. So it's a very simple assay. Well, he puts our molecules in and finds, lo and behold, that all of our SAR is shifted, because we end up screening everything now in his cells. And it turns out the open ring molecules are more potent. Now there, again, it's not night and day, but it's the difference between something that was 0.5 that's now 5 micromolar and something that was up in the 5s that's now down in the 1s. So shifting by a factor of 10, by an order of magnitude. So what you're doing could be system dependent. So if you're dealing in a model system that is not actually close to your disease system, when you finally get to your disease system, you may not be there. Okay, so at every stage, we have to validate and reconfirm using whatever is available. Okay? So he has two systems, right? He has cells, he looks to see how they grow, and you sprinkle the compounds on, and see if they live or they die. He also has animals. I'm not going to show you the animal data. This is not supposed to be a biology lecture. But he has animals that fluoresce when their lymphomas grow. No, pardon me. They emit light. Okay, it's luciferase system. So he can put them in a luminometer, and he can watch their tumors grow, and he can give them the drug, and he watches the tumors shrink. And so we can watch the tumors growing and shrinking in real time. So cells, animals, confidence level starts to go way up. So these halo substituted, it's interesting, if you don't have a halogen here, okay, and you don't have a methyl group here, you have no activity. You put a methyl group, activity goes up. You put a halogen, activity goes up. You put in two of them, and it's okay in this system. But we discovered some things. If you go to dihalogen, okay, things look good. If you go to a mixed chlorobromo, okay, now we're down to 0.5 for these two cell lines. One's germinal, one's not. I can't remember which is which. The methyl group, okay, now in the R position and in the R prime position, also well tolerated. It turns out you can't mix them. Okay, optimizing the methyl group, optimizing the halogens, doesn't work. Okay, can't do it. Can't start sticking a halogen over here, put a methyl group over there, and hope to get something more. Actually killed it. So the best to date would be the dihalogens, and the dihalogens are active in several models of animal models of lymphoma, human lymphoma. And this is just a slide that says what I told you. We looked at a variety of things. You could swap this out with pyrazine and imidazole. You could put the attachment one bond over instead of the 2 position and 3 position. That killed activity as we sort of work our way up. The alkylated thiophene were active, but it wasn't translatable. And in the end, this is where we need to be. It's the dihalogen compounds. So far, doesn't mean if we did more, we wouldn't find out something very interesting. We're limited to some extent by the chemistry. If we decide we want to make changes out here, it may be that we could modify this molecule. Maybe we have to go back to the beginning of a new synthesis and actually incorporate those changes from the beginning and bring it all the way back up. Okay. There was a paper in Nature just last week on a new method to take molecules that contain nitrogen compounds and stick some new substituent in with no antecedent. Just stick a carbon-carbon bond on one of these sites. That's not the stuff chemistry can do. That's why even though they were only getting 40% yield, it ended up in Nature as a full article. Okay? For the chemist, it's actually generally very arduous. Go back to the start, build it up from the beginning. So, typically we begin with an assay. and we develop a lead molecule. But it could be the case that our lead molecule comes from a high-throughput screening assay from a library, and we would go through a cluster analysis, figure out some cheap SAR, and move on from there. Or it could be the case that there's something reported in the literature, or there's something in the patent literature that you see, that you think, if I had this, if only I had this, I could test a hypothesis. Okay? Maybe it's not ideal. Maybe you still want to do high-throughput screening later. But if you can start with something that's really active, okay, it's generally easier to tinker with it than it is to generate activity from nothing. Okay? When you screen a library, a typical hit is 5 micromolar, 10 micromolar. That's where the limits are typically set. You get hits. You get something in the 1 micromolar range, you've done pretty well. Okay? Getting that thing down, sub-micromolar, getting it down, you know, if it's an isolated component assay into the 1 minute, could be a year. Okay, could be longer. So lead molecule. We try and identify the active domain. We cut off one portion. We cut off another portion. We try and figure out what's actually essential. What generates activity? What generates specificity? What's causing complications? It may be the case that there's a group that's highly metabolizable. If we get rid of that group, maybe it survives. Maybe we don't have a P450 problem. So functional groups, activity, selectivity, synthesis of derivatives. We can make pet labels, little things. Just remember, imaging work. We can do interesting things there. Molecular modeling may be appropriate if you know the target in detail. Usually it takes a crystal structure, something precise. We define the SAR, the activity, the selectivity, things that we typically don't do on a routine basis, but we have an LC-mass spec. We can do some cheap, i.e. not in preparation for human trials, but in preparation for animal trials. We can give you some ADME-tox data, okay, absorption, distribution, metabolism, excretion. It's possible with one of the companies to do ADME-tox modeling. And in general, sophisticated ADME-tox testing is a send-out. We usually don't do that here, but we will do some quick and dirty work ourselves. Acknowledgements. Our coming band of chemists led by Shizan Dang, Dr. Dang, likes to be called simply Dang. Milan Stojanovic, PhD organic chemist, is also involved in the project. The chemists generate data. The data goes into grants. The grants hire chemists. And so we'll have collaborations with others, with Jonathan Brash, with Tavio Arancia. And that's one of these here. And then these are some of our former group members who made important contributions to these two projects. Thank you for your time.
We've got time for a couple of questions.
So, let's say I run a screening facility, I get 3 hits, I come to talk to you, what happens?
We look at the 3 hits, and we see if one of them is Taxol-like, and we shouldn't touch it. It's too complex. Say ideally we find 2 hits that look plausible from a chemical perspective. We will search the literature and see what's known. Maybe there's something in the literature saying that this structure was actually looked at, and it kills, you know, every animal that got it. You know, we'll try and eliminate them based on what's in the literature. Okay? We will look at the patent literature and see if this series has been actively engaged in what's known about it. Okay? We will go to the chemical companies, and we will see if there are molecules available commercially that will allow you to do some quick screens and see if, in fact, you can get some cheap SAR. And then when we have all of that information, we'll make a decision on which one we're going to try. And then we will dissect it into its domains. And we'll make separate domains. And we'll try one, we'll try the other, we'll try the third. We'll connect 2, we'll connect 2, we'll connect 2. We'll try those and see if we can figure out what's the minimum portion we need to play with to optimize activity. And maybe you've got a counter screen. Maybe there's some activity you don't want. We'll be looking at that as well as we look at the components to see if maybe it isn't important for activity, but it's important for selectivity. And then finally, we will chart out how we might make this and how we might make it in a fashion that will allow us to do, you know, branched syntheses where we can substitute components and make 10 varieties of this and 10 varieties of that, and combine them and give you a 50 or 60 compound extravaganza. You'll test all of those, we'll get that data, and then we'll get another round of hypotheses. You'll have some standard that you'll want to reach, and when we reach that standard, the project's done. Things go well, and we're moving to animal models. There's a molecule you think is really great, we'll make a gram of it. If it works in a few animals, we'll give you 15 grams of it. You'll take those 15 grams, we'll publish a paper in Science.
I just add to that that because of this $6 million from NISTEM, that some of the chemistry work, as some of the screening work, will be subsidised through the grant. And so we're going to help the first projects, so some of the chemists are partly paid for this grant. Yeah, one second question.
Could you tell us, indeed, if you use the facility for these experiments, and if so, how? Because it seems sort of it's post-high throughput, most of what you said.
Right, all of this is, when you do a high throughput screen, you get a molecule that's not useful, generally. Okay, it's going to require some optimization. I mean, if you do a screen and you get a molecule that's got nanomolar potency, time for champagne, okay? That's generally not what happens. So it usually takes, it takes chemistry to improve the activity to reach the hit, to take the hit to a probe, and it absolutely takes chemistry if you want a new composition of matter. So if you want to publish on something using a unique molecule that is just, Columbia patented intellectual property, then you have to do chemistry. Because you don't own the molecules that come from the library.
But just to be clear, if you want to do a screen, and you haven't done it yet, you go and see Chuck, or Dan can also help with that, and the results from that screen, you'll either go and see Don and Dan, or Rashid and Brent, and you'll be hearing from them. So this is talking about after the screening, mostly. But all those things are available to you. So I think we should move on. Okay, thank you very much, Don, for making time. And we're very grateful to Perk and Elmer for supporting the day. And so Paul's going to tell us about some of their cell-based assays.
Thanks.
And then we'll have a coffee break.